Thank you very much for the introduction. It's a pleasure to be here. I actually have not talked to this particular crowd before, so if you have any questions or anything's unclear along the way, please feel free to just jump in. It's supposed to be casual, so don't worry about just raising your hand and saying that doesn't make sense to me. So no embarrassment, no shame in that. Furthermore, what I'm going to talk about today is pretty unfamiliar stuff to most people. So I'm going to talk about polyploids, which are genome duplicated organisms. So there's a lot of weird stuff that these guys do. So again, if something doesn't make sense, feel free. I'm happy to stop and clarify at any point. So as I understood it, the theme of this workshop was more like genomics, right? OK, so luckily, that is what we do. I feel like it's kind of an Alice in Wonderland time in evolutionary genetics. And the genomics revolution has really made that kind of possible for us. It's given us a whole bunch of new tools and technologies that we can use to better understand the genetic basis and the molecular basis of evolution and adaptive processes. So it's been extremely important in biology. It's extremely important in what I do. And it's also been very, very good for opening up non-model systems. So allowing us to kind of move beyond the Drosophilas and the Arabidopsi and things like that to actually explore some additional questions that are not possible in those systems. So I, like many other people in this place, am very interested in how genes translate into phenotypes of organisms. So this diagram is meant to be a chromosome on the left with a couple of genes on it. Those are just the colored blocks. And how they're conditioning the phenotype of these ravens, so what they look like, as well as how they are adapted to their environments. So there's a lot of crosstalk here. The environment is feeding selection onto the organisms, which in turn shows up as signatures of selection in genes. But there's a whole other layer of stuff here. And this is one of the main things that I'm interested in, is you can think of the genome almost as an ecology of genes. There are lots of genes here. They're interacting, they're cooperating, they're fighting with each other sometimes. But certainly, they all have to kind of work together to make cells function. And on top of that, they also have to be able to function properly within their cellular context. So it goes both ways. So this kind of forms the three sorts of questions that we're interested in pursuing in our lab, and mostly using these genomic tools to try to understand and get a handle on. So first of all, we do work on genetic adaptation to habitat. I'm not going to talk about that work today. But if you're curious, you can come talk to me about it afterwards. So we are interested in that sort of tree part of the equation. But we're also very interested in how genes adapt to genome change. Like, what happens? How does a gene respond when suddenly its whole context changes and the whole rules for it change? We're also very interested in the evolution of gene networks. I'm also not going to talk about that today. I'm going to focus on this middle question. So the species we work on is actually an Arabodopsis, but it's not the one that you might think of when you hear the word Arabodopsis. So that on the right is Arabodopsis thaliana. That's the fruit fly of the plant world. It's the one that molecular plant geneticists like to use to do most of their work. This is not the one we work on. We work on this one. It looks better. It smells better. It's much bigger. It's an obligate outcrosser, so it needs to be pollinated by insects. And that may sound like a fairly trivial detail, but it's actually really important for trying to understand signatures of selection in the genome, for understanding evolutionary processes. It actually makes it hard if something is self-fertilizing, because it kind of breaks a lot of rules, and selection can't operate as effectively. Overall, this genus is very user-friendly. So this is just a very schematic phylogeny of the genus. The branch lengths are not really correlated with time, so I apologize, it's not a very accurate phylogeny. This is Arabodopsis thaliana out here. So again, this is the model system. It's been sequenced. There's a genome browser available and all kinds of genetic tools. And then we come up here. Arabodopsis lyrata is a related species. It's also been sequenced, and there's also a lot of genetic and genomic tools available. And the one we work on is up there, Arabodopsis arenosa. It's about 2 million years at most diverged from lyrata. So we have a lot of tools available by being able to compare to this related species for which the sequence is already available. We have sequenced arenosa ourselves, but it's not quite as complete a sequence as is available for these other two. So we use those as our references. One of the main questions that we're interested in is genome duplication. And Arenosa, the species we work on, does have populations that are genome duplicated. And I'll get back to that in a moment about why that's important. But first I want to talk about what that even is. So when genomes duplicate, they give rise to polyploid organisms. These are organisms that carry more than the normal 2 homologs of every chromosome, right? You and I carry 2 copies of every chromosome, 1 from mom, 1 from dad. These guys get 2 from mom and 2 from dad if they're polyploid, if they're tetraploid. So if they have 4, they can have more. So these kinds of events are thought to be very important in evolution and have given rise to speciation and plant and animal diversity. So this is a crude phylogeny showing the plants up here appropriately in green, and the animals in blue, and the fungi in orange. And it's just representing different genome projects. So these are all sequenced genomes, sequenced organisms. And then the red dots are spots on the phylogeny where we think that genome duplication events have happened. And the red ones are the ones that we're really certain about. And the orange ones are ones that are inferred. So these are inferred from extensive bioinformatic analyses of the genomes. But they happen so far back in time that we can't be absolutely certain that that's really what went on. But that's what we think went on. This is not my work. I pulled, actually, this is redrawn from something I found on Wikipedia. So this is all published work from other groups. One thing you may notice about this is that there are a lot more of these in plants than in animals. And that's actually real. That's not just a bias from this chart. It seems to be much more common in plants that genomes duplicate. But it does happen in animals, and there do seem to be events at the base of the vertebrates. So we're part of the vertebrates, obviously. So that is potentially in our lineage. And then in amphibians and fish, there have been several events. There have also been several events in the insects, which are not illustrated here. So here are some examples of genome duplicated animals. So it's known from tree frogs as well as xenopus frogs, which you may have heard of. They are also a model system, particularly for development. Salamanders, so another amphibian, brine shrimp, and then salmonid fishes and carp and their relatives like goldfish. It's also worth noting that polyploidy is actually also a normal part of development. So you have polyploid cells in you right now. So there are polyploid cells in your liver, there are polyploid cells in your bone marrow, and in some of your nervous tissues. So this is a normal part of development, and that's true for both plants and animals. But interestingly, cancer cells are also often polyploid, and they somehow become unstable. They start losing chromosomes and get all wacky, and that's called aneuploidy, when they start losing chromosomes. So something goes wrong in cancer cells to this sort of fundamental developmental process. And how that works, we don't fully understand. Plants. Whole genome duplication is extremely common in plants, and it has contributed to a lot of species radiations in plants and a lot of diversity. And in a lot of cases, it allows plants to colonize new habitats that were not available to the diploid progenitors. So it kind of expands their niche and their evolvability is one way you can think of it. A lot of our crops are polyploids. So chances are you ate 1 or more polyploid individuals today. I had a banana for breakfast. That's a triploid. Potatoes are tetraploid, which means that they have 4 copies of every chromosome. Remember, you and I have 2. Wheat is hexaploid, so it has 6 copies of every chromosome. Strawberries have 8. And the champion among crop plants is sugarcane, which has 12 copies of every chromosome. It's dodecaploid. So this is pretty remarkable. I think it's remarkable that these organisms work, that they can survive, that they can figure out what to do with 12 similar chromosomes. I mean, how that works is amazing. Yeah?
Is the polyploidy something that occurred after they were cultivated, or is it something that occurred before they were cultivated?
That's a great question. So the question was, for those of you who couldn't hear, are these polyploidy events pre or post domestication? So are they associated with our activity cultivating these plants? Actually, for potatoes, that's not the case. So their wild ancestor is also tetraploid. For bananas, the ones we eat are actually hybrids between a wild tetraploid and a wild diploid. So that wild tetraploidy event was not related to the domestication, but the fact that the bananas we eat are triploid is related to the domestication. The hexaploid wheats were derived from wild diploids. And through various domestication events, they became first tetraploid and then ultimately hexaploid. Strawberries, it's post-domestication. Sugarcane, it was also a hybridization event from lower ploidy progenitors. So the fact that it's dodecaploid is related to the domestication, but it came from hexaploid ancestors that were wild species. But it does actually bring up an important point, because polyploidy often makes cells bigger and often makes fruits bigger. So you will often find it associated with domestication events. So thank you for bringing that up. There are actually two major kinds of polyploids that are actually different in terms of the challenges they face and the kind of biology that they have. So on one hand, we have autopolyploids, and these are things that are duplicated within species. So you go from having 2 homologous chromosomes to having 4 homologous chromosomes, but they're all derived from the same sort of gene pool. So they're all roughly equivalent. In contrast, allopolyploids are associated with hybridization as well as duplication. So 2 different genomes are coming together. They're usually not hugely different, but they are definitely distinct. They come together and then duplicate. So what you end up with is indeed 4 roughly homologous chromosomes, but there are 2 that are more similar to each other, 2 sets of 2. And this is actually quite different, because this means that these guys essentially have duplicated all their material. These guys have also, but they actually segregate randomly, so it's not really strictly duplicate genes. This is the kind of situation we're dealing with. So Arabodopsis arenosa is an autopolyploid. It's a within-species duplication. This is also, for example, what like cancer cells would be. They would be a within-species duplication because they're happening within your body. So it's the set of 4 chromosomes. And this is particularly challenging for example, the meiotic machinery to deal with. And I'll talk about that, I think, in a moment. Not quite the next slide. So these have great problems during meiosis. These have more problems with things like gene regulation, like figuring out how to regulate all that extra stuff. And it turns out that probably has more to do with the fact that they're hybrids than that they're duplicated. These guys have more of the problems associated strictly with duplication. So after polyploids first form, which in plants they usually do by unreduced gametes coming together, and by unreduced gametes, I mean gametes that have not cut the number of chromosomes in half during meiosis. So they go through meiosis, but at some point all the chromosomes go to one pole, and you end up with a gamete that has double the number of chromosomes that it's supposed to have. This happens in humans actually fairly commonly. It's thought that about 15% of spontaneous abortions in humans are due to polyploidy. So it's actually not that uncommon, even in us. We just don't make it. We don't survive if this happens. But in plants, they can survive. But these early arising polyploids often have all kinds of problems. So they have extensive genome changes, rearrangements, insertions, and deletions of genetic material, and what we call genomic instability. They can actually lose entire chromosomes or break chromosomes in half. There's all kinds of chaotic stuff that can happen, and this is very, very challenging for them, and usually they suffer a fertility penalty as a result. But there are some lineages that have enough fertility to make it, so they can survive. And if they have other advantages, for example, if they're able to colonize new habitats, they can actually find a new niche and survive and make it in the world. And as they become established, what we see is increased meiotic stability, so they start to figure out how to sort those extra chromosomes properly. They become cytologically diploid. I'll tell you in a moment more about what that means. And they have extensive evolutionary potential. It turns out they have a huge ability to colonize new habitats and to radiate into new forms. There's actually quite a lot known about early events in polyploidy, but virtually nothing about what we could consider a sort of stabilization phase. How do you get from this problematic situation to this really stable situation? What are the genetic events? What's happening to the genome between those 2 points? And that's the question that my lab is really interested in trying to figure out. And again, there's virtually nothing known about it. And the reason that virtually nothing known about it is actually a tool problem. So polyploids have been of interest to people for decades. It's not that people haven't been interested in this question. It just hasn't really been accessible to try to understand what's going on. So their genome sequencing has really helped us, and that's the project I'm going to tell you about today. So there really hasn't been much exploration of the possibility that polyploids may genetically adapt to their own genomes. And that's the story I'll tell you today. So this is the distribution of Arabodopsis arenosa, again, the species we work with. The blue dots are collections that we have made that are tetraploids. So these have 4 copies of every chromosome. And the red dots are collections that we have made that are diploid, that have 2 copies of every chromosome. And this is the material we're comparing. These are the people who, in addition to me, made this collection. The range of the diploids and the tetraploids was actually characterized by another group some years ago, but our material seems to fit with this. There's a very narrow point where diploids and tetraploids overlap, so where their geographic range overlaps, but nobody's actually ever found a population that's mixed ploidy. So if you go to this region, you'll find them in isolated patches, but never will a patch have both diploids and tetraploids. They're always either all tetraploid or all diploid, but you can find mixtures in that region. Everywhere else, you'll find only one or the other. Okay, so I wanted to briefly go over why meiosis is challenging for autotetraploids, like arenosa. And the reason that I want to go over that is because that's what we're mostly focusing on right now, is how did meiosis adapt as a process to this genome duplication event. So this diagram is actually meant to illustrate diploid chromosomes. So this is homologous chromosomes from a diploid after duplication. So the sister chromatids are present. That's the double strings here. And then these are the 2 homologs, the pink and the brown. As they go through meiosis, they recombine with each other. This is straight out of the textbooks. And when they pull apart at anaphase, you end up with 2 meiosis I products that are recombined versions of those original homologs, right? That's diploid meiosis. So what happens when you think about a tetraploid? So suddenly, this guy has 4 homologous copies of these chromosomes instead of 2. So what's going to happen? And one thing that's known from extensive cytological work that's known to happen is that you can get crossing over events onto all 4 of the homologs. So they can all kind of get tangled up in each other. And this is called a multivalent. So whenever you see these kinds of crossing overs onto the different possible partners that tangles the whole mess together, it's a multivalent. And as this starts to pull apart at anaphase, so the 2 centromeres start to go opposite directions, you can see that there are lots of tangled bits in there. It may work out just fine, but in many cases it doesn't work out just fine. You'll get chromosome breakage, you'll get chromosome losses, or just pieces of chromosomes lost. So it can be very problematic, isn't always. So these kinds of multivalent configurations are very commonly observed in newly arisen tetraploids, wild ones, as well as ones that we can generate in the lab with chemicals like colchicine, which is a spindle inhibitor and can generate polyploids. This is a problematic configuration, but this one's even more problematic. So this is called a trivalent and univalent configuration. So what happened here is these guys recombined in such a way that they left this one out of the game. And so this one will actually segregate randomly now at meiosis and will lead to some gametes that have an odd number of chromosomes. And these guys will have trouble because they have 3 centromeres, so 2 of them will go to one pole and 1 will go to the other, but it's random which pole gets 2 and which pole gets 1. So this can be a real mess, real problematic mess. And those are common also in new polyploids. But when we look at established, nicely stable, evolved polyploids, this includes our crops as well as the wild species that we find that have been around for a while, you'll find that they form exclusively what are called bivalents. So cytologically, these look exactly like diploids. You would never guess from just looking at the chromosomes that they have 4 equivalent copies of these things. They always go in 2 and 2, like this. So there are actually 2 different ways that you can ensure this to happen. And if you think about it, they make actually perfect sense. And this is indeed how these organisms do it. So in one situation, you can have a pairing partner preference. So if the blue chromosome always pairs with the pink one, and green always pairs with brown, then they're always going to form only bivalents because they will never want to pair across this set. Does that make sense? Okay. The other way you can do it is limiting crossover events. And this will make sense if you look at this guy. So if you allow one crossover event to happen on this chromosome, but then that prevents the second one from happening anywhere on the length of this thing, then there's no opportunity at all for it to cross over to one of these others because it's limited to that one. And that will also lead to stable bivalent formations. And both of these processes have been shown to work in established polyploids. This first one is much more common in allopolyploids, where you already had some differences between the chromosomes, and all you need to do is really to firm up those differences. So wheat, for example, is an allopolyploid, and it's been shown that it operates in this way, that there are preferences for pairing with particular chromosomes. On the other hand, it's also been shown that in autopolyploids, so these are the ones that have 4 equivalent homologs, copies of every homolog, that it's actually more this mechanism, that they tend to limit the number of crossovers to become meiotically stable. So what about this weird species that we work with? This is a diagram showing what some of these chromosomes might look like cytologically. So above this dotted line, we have bivalents. So these are the diploid-like ones that are meiotically stable. And below this line, we have multivalents. So these are the configurations that I was talking about where all the homologs are crossing over onto each other at essentially random. And then unpaired univalents. So everything below this dotted line is fairly meiotically unstable. And everything above it is stable. When we look at arenosa, so this was actually done by one of our collaborators in the UK, they find 8 blue blobs. And the reason, 16 blue blobs. The reason that we're looking at blue blobs is because they're stained with a chromosome stain called DAPI that lights up blue when you shine UV light on them. So that's what's happening here. Arenosa has 32 chromosomes. So the fact that we're seeing 16 blue blobs means that they're forming bivalents. They're not doing this down here. Each of these right here is a configuration like this or the one at the top that I can't quite reach. And these ones, that one in the middle where there are 2 chiasma, are these ones that are kind of plus-shaped. So if you're used to cytology, which I'm not, I needed collaborators to do this, they can read these things and they can tell us what the configuration is of these chromosomes. These ones that look all strung out, that's this kind of situation where they paired at their very tips. But the important point really is that we're not seeing these multivalent configurations. We're not seeing this unstable stuff. So this is a meiotically stable polyploid. This is consistent with findings from another group, which found a similar result with a different strain, not the one we're working with. But there's another interesting point. So this white arrow is showing this one little blue blob. This is actually univalent. This guy's unpaired. And it turns out that this parent plant had 31 chromosomes, not 32. So it's actually missing one. So there is a little bit of indication that there may be a little bit of residual instability, but it's not extensive. It's not like they're all forming multivalents. So my lab decided to take a genetic approach to trying to understand what's going on here, trying to understand how this thing became a stable polyploid. So we took a genome resequencing approach. You may have you've probably heard about these already today, right? No? Okay, well then I'll go over it. So what we did was we went back to our collection and we sampled 12 individuals from 4 populations. And that would be sampling 48 alleles, because these guys are tetraploid, of every given locus. We sampled 2 railway populations and 2 outcrop populations. They come in different habitats, and we wanted to make sure that we weren't sampling habitat adaptation instead of adaptation to polyploidy. We've also sampled 2 diploid populations from the Carpathian Mountains in Slovakia. And those are the white circles up at the top. So what we've done is we've actually sequenced these tetraploids completely. And you can do this nowadays with what's called short-read sequencing. Very easy, very cheap. Well, it's not super cheap. Can't do it on like a child's allowance, but it's gotten a lot less expensive than it once was. And so it has become possible for us to do these kinds of experiments, just sequence the whole genome. Why not? So we sequenced these 12 and those 8 diploids. And then we did two kinds of genomic analysis. So we did a genome scan, which I'll tell you about in just a second. I have a slide explaining what that is. Looking for selection. So we're looking for evidence that particular genes have been under natural selection to stabilize this genome and make it be able to deal with the fact that it has four copies of every chromosome. And then we did a second one where we actually compared. So this first one is looking only at the tetraploids compared to one of their diploid relatives. And this one is actually comparing the diploid pool with the tetraploid pool and looking for genes that are different. So where we have a different version in the tetraploids than we have in the diploids. So the first thing that happens in one of these short-read sequencing approaches is we make lots and lots of bits of sequence across the genome. These are 100 base pair fragments. So they're very small compared to the size of the genome. But we make millions and millions of them. And then we assemble them onto a reference genome. And what I mean by that, in our case, we're using Arabodopsis lyrata. Ideally, it would be nice if we could just assemble them de novo. And you can do that, but not for an outcrossing tetraploid. It turns out that's just impossible with today's computational power. It would need like a mega cluster. So we actually just pile them onto a reference genome, and the sequences just get put where they are the most similar to this reference genome. It does allow polymorphism, though. And then we can scan through this sequence and look for all the sites that are different, either among individuals or between those individuals and the reference. Is that cool so far? OK. And then what we can do is we can make plots that look something like this. This is just a schematic one I drew up. It's not actual data, but our actual data looks very, very similar to this. So this blue line going across the bottom here is the reference genome. So that's meant to just pretend that's a DNA sequence from Arabodopsis lyrata. And then what we have going up here on the y-axis, each of these dots represents a site where at least one sequence from arenosa is different from lyrata, from this reference that we're comparing to. And the higher they are up on this chart, the more of the arenosa sequences are different from lyrata. So we call these high-frequency polymorphisms and low-frequency polymorphisms. Most of them are low-frequency, rare variants. But there are some that are more common, so where like 40 out of the 48 arenosa sequences are in one state that's different from lyrata. So that's indicated there. These are the ones where maybe 1 out of the 48 sequences is different, and that's where maybe 48 out of the 48 are different. That would be called a fixed difference. And then we just kind of scan across the genome with a computer, of course, not with our eyes, and look for regions with excess polymorphisms, or single nucleotide differences, that are at high frequency. So we look for regions that have too many of these compared to the genome-wide average. And that's an indication that a genetic region may have been under evolutionary selection. It's one of the signatures we look for. It's one of the signatures they look for in the Human Genome Project. This is very common, and this is not specific to plants or polyploids. This is universal. And they do the genome scans in the same way, actually. Most of the techniques that they use, we borrowed and adapted to polyploids. So one of the kinds of tests we do, I'll walk you through this because this is kind of complicated, is to look for an unusual site frequency spectrum. So on the previous slide, I was showing you the differences in the frequencies of these polymorphisms that we can look at, right? So this is basically just a histogram plotting out for a single gene what those frequencies are. So down here, we have polymorphisms where only 1 out of the 48 arenosa sequences differs from lyrata. And then it's a proportion. So we now know, so the blue is silent sites, amino acid changing sites, and intron sites. We now know that the majority of these are rare. So out of all of the polymorphisms in this gene, something like 70% of the polymorphisms are found in only one of the arenosa sequences compared to lyrata. And stuff out here would be things that are very different, so where 48 or 40 or whatever sequences are different. So this was a gene that I picked out from the genome average. So this is an average gene. This is what you would see randomly sampling the genome for the most part. This is an outlier gene. Can anybody see what's different about this bottom panel from the top one? Yeah, there's more weight down here, right? There are more things over here. That's exactly right. That's really what we're looking for, is an excess of these kinds of high-frequency polymorphisms. So the test that we do actually has a rather complicated way of assigning scores to these histograms. And it has a very good power to detect ones that have these outliers on this end of the spectrum. That's really what we're testing for. And then we can do things like take the top 1% or the top 5% or whatever cutoff we want to assign and look at those genes. And this is just an example of what we find, so I don't expect you to read all of these genes or understand what they are. It doesn't matter. The point is, we get lists out. We get lists of genes that are potentially under selection, and this gives us nice candidate genes that we can then follow up on. And we take these as hypotheses. This is not proof that any of these genes are under selection, but it's a hypothesis. So it can guide us to genes that may be under selection. And then we have to do additional tests to see that they actually are and try to understand what functionally has changed about those alleles in those particular genes. So this is just a list showing some of the genes that we found that are related to meiosis. And interestingly, given what I was telling you earlier about how autopolyploids generally stabilize meiosis by reducing recombination, a lot of these genes are implicated in recombination, in how much recombination there is between chromosomes. So it makes sense. So it's nice. These are really good hypotheses now. They have sort of a functionally relevant explanation and something where we can go back and now have something to test. So we can go back and ask whether, in fact, these different alleles that are found in tetraploids and diploids are actually having an effect on recombination in these plants. And that's what we're working on testing now. I wish I had data for you. I don't yet. But it really does point to the idea that when we were comparing the sort of unstable new polyploids to the stable polyploids, that they are doing it by this mechanism, by this limiting crossovers rather than pairing preferences. What was also interesting was that there are really cool parallels with other kingdoms, including us, and I'll get to that in 1 second. There was a paper that came out some years ago where they did a study in yeast that I would have never thought would work if I had heard about it ahead of time, but it worked, so luckily they didn't talk to me beforehand. They tested about 3,000 mutants in yeast for their effect with ploidy differences. You can make yeast diploid or tetraploid. So what they did was they took these 3,000 mutants, tested whether they had an effect in diploid yeast, and then tested whether they had an effect in tetraploid yeast, and asked whether there were any that were particularly troublesome for tetraploids but not diploids. And they, in fact, found 39 genes that are lethal to tetraploids and irrelevant to diploids. 39 genes. The interesting thing was that these all implicated what's called genome stability. They called it genome stability, homologous recombination, sister chromatid cohesion, and spindle function. And these are exactly the functional classes that we're finding under selection in Arenosa. So it suggests that there's something fundamentally shared between this fungal system and this totally different experimental context and the plant system that we're working with. And that shared feature is probably the fact that they have duplicated their chromosomes. We also find several actual homologous genes, so where we can say this gene is the same as this gene in yeast, that are on both lists, that are on their list as well as our list. What's also been fun is that several of these genes are actually also implicated in human diseases. So a number of them are oncogenes, which means that they're implicated in cancer, and particularly in the instability of polyploid cancers. So that's why I mentioned the cancers early on, is these genes are actually involved in that instability. So they're known to contribute. So if you have one allele of one of these genes, you may be less prone to having one of these unstable polyploid cancers than if you have a different allele. So they've been implicated in that in humans. So that was exciting to us because it suggests that the polyploidy that's happening in cancer cells may not be all that different from what's happening in the plants, and that we can actually learn something from studying these systems in different kingdoms. And in plants, unlike in polyploid cancers, we can actually study how the genome can evolve to adapt to that state. They're also implicated in genome instability syndromes such as name, make, and break syndrome, Bloom syndrome, and Cornelia de Lange syndrome. and Alzheimer's and several neurodegenerative diseases in addition to Alzheimer's. One of the first genes that we've started working with is ASY1, which is a gene that controls synapses formation, which is the first step in recombination. And this is just a plot showing polymorphisms. It's similar to that genome scan plot that I showed you in the beginning, where these are just different nucleotide polymorphisms relative to the reference across this gene. And the red ones are amino acid polymorphisms. So this is where the actual protein sequence has changed. And right in the middle of this highly conserved domain that's shared between plants, fungi, and humans is an amino acid change that's at high frequency in the tetraploids and low frequency in related species. And it does similar things in different plants. But interestingly, in polyploid wheat, they've actually quantitatively modulated the levels of this gene and showed that it does affect pairing. So they can get them to go anywhere from just forming univalents all the way to these complex multivalents just by modulating the levels of expression of this one gene. But it's not under selection in wheat, but it is under selection in arenosa. And so we're very excited about that and think that that may actually have something to do with it. So we're working on that. One of the problems with our test, however, was that everything I've been telling you about is comparing Tetraploid arenosa, these guys, with Arabodopsis lyrata. But there's something in between. So when we look at this and we can say, well, they're different between that guy and this guy, we don't actually know whether they happened on this part of the branch or this part of the branch. This part of the branch would be relevant to ploidy, but this part would not. So one of the first things that we wanted to establish was that it was actually happening on that top bit. This was done by an undergrad in my lab, and she figured out that luckily and fortunately, it is apparently under selection in this part, specific to polyploidy. So this is just showing a pie chart of the frequency of the ancestral allele, so the one that's found in all these other plant species, as well as all the way out to fungi, actually. It's at about 95% frequency in the diploids. And when she tests tetraploid alleles, she finds the alternative version in about 90% of the cases. But interestingly, you do find the ancestral allele in the tetraploids, rare variation. And more importantly, we're finding the derived allele in the diploids. And this is exciting because it suggests that this thing may have been selected from variation that already existed in the diploid. So it may have been sort of pre-adapted. It may have been sort of preliminarily already capable to evolve this new allele because it didn't have to do it from a new mutation. It was already there. It was probably neutral, just kind of floating around, and then selection could act on that. And that's a much easier way of getting selection to be effective. It can work very rapidly and very well when there is so-called standing variation. Interestingly, ASY1 has a partner, and we see the same pattern for that partner. I won't go into the details of that, but there's another protein that does the same thing together with the ASY1, and it's showing the same pattern. There's not much to see here. This was really just an alignment of this part of the protein. And the point I'm trying to make here is that this amino acid is highly conserved. So when you look across plant diversity, most plants have lysine in that position, which is the K. Some have switched to arginine, which is that N independently. The vast majority have a K. Fungi, yeast have the K. And the tetraploid arenosas have an E, which is unique. All right, so now I'm going to summarize what I told you. So importantly, we figured out that arenosa has evidence of having genetically adapted to genome duplication. And this was actually really fun for me because when I first started my lab, people told me I was insane for doing this project and that I wouldn't find anything. They said it would all be about proteins and sort of changing the structure of the chromosomes and that there would not be genetic adaptation to polyploidy because you can't adapt these genes, is what they said. They're too fundamental. So I'm happy to say that fundamentally important genes do seem capable of evolving. There's no evidence of a genetic bottleneck associated with polyploidy. It's probably not that important for you today, but many polyploid species actually arise from single individuals That does not seem to be the case here. It seems that there were multiple progenitors, multiple individuals, not multiple species, just to be clear. Meiotic chromosome in particular seems to have been under selection in these guys. But likely there are also other important processes. So I haven't told you about the rest of that list. There's a lot of other stuff on that list. And we are starting to work with trying to understand what it means. I don't want to leave you with the impression that meiosis is the only thing that had to change. There's plenty of other things that had to change. We're just not working on them at this point. And again, some things may have been selected from so-called standing variation. Another interesting point is that actually many interacting genes are under selection. So a lot of times if a protein has to partner with another protein to have a function, if the first protein is under selection, a lot of times we'll see a signature indicative of selection in its partner as well, which is cool. It suggests that they have to kind of co-evolve with each other. And that's really common. We've seen lots of partner pairs on our lists. So these are the folks who did the work. So primarily the genomic work was done by my graduate student and postdoc, Brian and Jesse. And then the ASY work was primarily my undergraduate, Catherine. So we welcome young students. We've actually also had some fantastic high school students. Kirsten Bomblies was fantastic, and she did a lot of work on, particularly on our work in habitat. So we do welcome high school students as well. A number of other people, collaborators and so forth, have contributed. And then finally, just to show you that scientists don't do only science, my postdoc wrote what might be the only haiku in existence about polyploids, but it does kind of sum up the point. So, all right, so with that, I'm happy to take any questions, or you're probably all tired and hungry, but yes.
So, I always thought that polyploids, except for plants, mainly plants are okay, but in animals, that polyploidy was harmful, would resulting in mutations and harmful changes for them. But this doesn't seem to be the case.
So there are far fewer polyploid animals than plants, and it does. Well, at least in humans, there are many cases where spontaneous abortions are due to polyploidy, so it's clear that we have problems with it. But it is clear that it has happened a few times in animals successfully, and why it's sometimes successful and other times not is actually really unclear. We don't fully understand what's going on, why some lineages can make it as polyploids and others cannot. There are some really fundamental differences in cell biology that may have a big effect on chromosome segregation, and those are the centrioles. So centrioles are little organelles that make the spindle that pull apart the chromosomes, and when they duplicate, as a cell duplicates, you can get all kinds of problems, and that's why polyploid cancers are unstable. So it may be that if you're an animal and you duplicate all your chromosomes, but you don't duplicate your centrioles for whatever reason, that you're just fine with that, and that you can be a stable polyploid as a result. Plants don't have centrioles, so they wouldn't run into this problem. So it could well be that that kind of fundamental difference in biology may contribute to the fact that it's easier to be a polyploid plant than a polyploid animal. They both have problems with it, but animals seem to have a much, much harder time, but it's not impossible.
Is there an evolutionary advantage to polyploidy because you have more opportunities for crossing over?
Oh, that's a good question. So the question was whether there's an evolutionary advantage to polyploidy because you have more opportunity for crossing over. There's certainly an advantage to having more genetic diversity, and they do have more genetic diversity. Whether it's because of crossing over is actually not known. I think it's a good question, but we don't have the data to answer it at this point. So, yes.
When you have a polyploid, do the genes of the polyploid chromosomes do the same thing still? Or is it that one of the genes might be under less control so that it can actually incur greater mutations and start having its own course?
Yeah, great. Yes, so this actually gets to one of the differences between the auto and the allo polyploids. So the auto polyploids, because they don't tend to have chromosome pairing preferences, at any given locus, they just randomly segregate 4 alleles. So there, the genes don't actually evolve as if they're duplicated, because they can still become homozygous, so deleterious alleles are still deleterious. But you do bring up a good point, because in allopolyploids, where the chromosomes do have pairing preferences, you can have one copy that's still doing the ancestral function, and then the other one is kind of free to evolve new functions, and that is seen all the time. That is very common. A lot of times the second copy will decay, but other times there are cases where they have evolved new functions or subtly different functions, or they partition the ancestral function across the 2 genes so that now you need both of them to have the full function. There are cases of all of this. So, but yeah, thanks for bringing that up. There was another question back there.
Yeah, I was wondering how all these copies would coordinate which gets expressed and which gets switched on and off.
Oh, gosh, you guys are asking such great questions, all of you. So for allopolyploids, that's a much bigger problem, actually. For autopolyploids, we have no idea what happens to gene expression. I think it might just be globally tuned down so that all 4 copies are expressing a little bit less, and that would be fine. For allopolyploids, where you have kind of different copies, it's actually very problematic early on when they form, and you do see all kinds of changes in gene expression and changes in chromatin structure that regulates gene expression. It kind of goes chaotic, and it seems that it sorts itself out because of selection just kind of selecting for the survivors. And eventually you can have kind of what I was talking about just now with the previous question. You can have divergence in expression patterns across the 2 duplicates in those cases. Yeah?
Does mitochondrial DNA become polyploidy in the polyploidy organisms, or
No, it does not, actually.
ever?
Well, no case that I know of. So because polyploids tend to be formed by unreduced gametes they're not reduced for the nuclear genome, so they have double the number of chromosomes there. But the cytoplasm, the mitochondria are just kind of, they're on their own clock. They replicate in their own time. And so the polyploid will inherit sort of the normal number of mitochondria from its mother that it normally would from a gamete. But it doesn't get any from its father, again. The cool thing about that is we can usually tell who was the mother in a polyploidy event. If it's an allopolyploid, we can tell who was the mother because of the mitochondria. But they don't have any kind of duplication. Yeah.
So I'm thinking about the connection between the evolutionary process within a plant where the generational effect is between this plant and its progeny, and then the linkage you make to cancer, where is the generational change occurring sort of after each cell division? Because we don't have cancer doesn't grow up and have children and grow up and have children, so we can't look forward as successive individuals necessarily. So is that whole evolutionary process compressed within the life of a cancer body?
So one interesting thing about these unstable cancer cells is it actually turns out that the aneuploid cells, the ones that have lost some chromosomes, are particularly aggressive. They're very good at growing, they're very good at dividing, and they're very good at ignoring any kind of cell division suppression signals from outside. The big difference between the polyploid plants and the polyploid cancers, obviously there are many differences, but they're not adapting to that polyploid state. There's no pressure for them to be stable. In fact, they do better when they're unstable as cancers. So from the purely selfish perspective of that cell doing well, it actually has no evolutionary incentive to be stable, because it's actually going to divide a lot more rapidly if it's not, ironically. You would think it would just kind of crap out, but it doesn't.
So the selection is for the unstable.
Yeah.
Yes, I see what you mean. It's not really selection, but the unstable ones.
It's not heritable selection in the sense of that we think of it passing on to progeny. It doesn't. But it's selection on that cell lineage, to be sure, to be actually highly proliferative. And then there's actually no advantage to being stable because you're more proliferative.
And they have to evolve into an unstable condition as opposed to reverse, which is
Yeah, yeah. Right, so there's no benefit to them to figuring this out. There's benefit to us to figuring it out and finding ways to treat cancer, but for them, there's no incentive. Yeah, there's one more.
So this is a fundamental question that goes back more to just the mechanics of meiosis.
Yeah.
Okay, and only because you brought it up before, so I have to follow it through. When we talk about the centrosomes, and of course the animals have centrioles and plants don't,
Yeah.
and yet both have microtubules and both set up a metaphase plate and move the chromosomes apart. Can you clarify for me, because I've been asking this at a lot of places and nobody ever can. So how is it that plants actually pull apart their chromosomes during metaphase when there is nothing essentially to be at the spindle poles?
Right, no, that's a good question. They do form spindles, obviously, you know that. They seem to start growing out from particular points on the membrane, like the plasma membrane. What exactly is happening there to organize it is actually not entirely clear, but it's thought that there are actually protein complexes that move to that part of the membrane and start the process of the spindle growing out.
So is there still a movement that, like, when you talk about prometaphase and...
No, actually there's no movement. They start from one point and they stay at that point and they just grow together, pull the chromosomes apart, and that's it. And then they just fall apart again and you can no longer see anything at that point. So there's no organelle that has to replicate. It seems that the proteins are kind of always there and then they just move to the right point and start spindle formation at that point.
In the case of cell signaling that we don't really understand what initiates the start of those microtubule organizational.
Right, like what causes them to grow. At least I don't know it. It may be that it's out there somewhere in the literature, but I don't know what that signal is.